Okay, so another biotechnological technique is called gel electrophoresis. And this is where you're taking a sample and you are pulling it out based off of size and charge. Polarity is also involved, but we can talk about that in a little bit. So size and charge. You do need something called an agarose gel, and that's going to sit in a chamber that is charged. You're sending an electrical current through buffer. So let's say that there is buffer. Within this chamber. Let's pretend this is your chamber and you have your gel. Okay, so you have your gel. These are going to be your wells. Okay. So your chamber here has electrodes on it. And that's going to allow a charge to run through. Now, depending upon what you are running, whether it's DNA, RNA, protein, or compounds, right? Some certain food colorings, depends on how you want to orient your agarose gel. Now remember, this is going to be here. This is your polysaccharide gel, agarose gel, typically made out of seaweed, okay? And so you're running through this chart. Now let's pretend, just for the example, that we are running samples of DNA. DNA, because of its sugar phosphate backbone, is negatively charged. You want to run your gel in the opposite direction. Opposites attract. And so you want to make sure that your charge is negative here, but positive here. So that electrical current is going to run through. And let's say you have your vials here of your different samples. And let's say this is sample 1. We'll do different colors. Sample 2. And sample 3. Gel electrophoresis is a great way to compare samples of these fragments to see if they are similar size. Especially if you're using DNA, you're not going to compare charge because DNA all has the same charge, but then you're looking at the size. So as you load up your gel, so let's say you put in sample 3 here. You're going to put in sample 2 in that one and sample 1. You turn on the machine and it starts to run your samples. After a certain amount of time, your samples are going to be maybe in different positions on your gel depending on their size. So let's say sample 1 was here, sample 2 was here, and sample 3 ended up being here. Now, what does that mean? Sample 1. Sample 1 is actually going to be small when you compare it to sample 2 and sample 3. So this one is the smallest. Sample 2 is going to be large. It actually gets stuck in that agarose gel. You can think of that. And so the larger the fragment, it actually travels less. And so then for sample 3, we'll say it's medium, right? So this one here is smaller than 2, but larger than 1. The other thing I did want to mention is, again, and this has to do with DNA. So if you're running DNA on a gel, it needs to be cut with restriction enzymes. Restriction enzymes will go along and look for particular patterns on a string of DNA. So let's say this is our DNA. And these are our nitrogenous bases, so base pairs. You can eventually make them pair up nicely, right? So our restriction enzyme is going to find a particular pattern, and there are many different restriction enzymes out there, depending on what you are looking for your fragment of DNA. So you find a restriction enzyme, it looks for a pattern, and it will cut your DNA in that particular way. So sometimes it will cleave it like this, or sometimes it's going to cleave it like that, leaving these what they call sticky ends.